Good morning. We finished up with sodium regulation in our last lecture, and today we will be finishing up ion regulation with a look at potassium and calcium and phosphate. Then we will be looking at how chemical buffers work and the systems that are involved. And then we will move on to respiratory regulation of acid-base, followed by renal regulation of acid-base. And then next lecture, we'll pick up with disorders of acid-base. So this graphic is where we ended last time. It is looking at the mechanisms that regulated sodium and water balance, and then sodium and blood pressure. So remember, those are kind of two separate systems, and they are covered in the previous lecture. For potassium balance, so for the remaining ions, we have potassium, calcium, and phosphate. I'm going to explain why those three are important. So what do they affect? And then what hormones are going to control their reabsorption or their secretion, so their regulation. So for potassium, calcium, and phosphate, the two items that you need to know for those three are: why is it important to regulate potassium? Why is it important to regulate calcium? Why is it important to regulate phosphate? As well as what hormone is the major regulator of potassium balance, calcium balance, and phosphate balance. Okay. For potassium balance, the reason why it is important to regulate potassium levels is because potassium is the main intracellular cation. Sodium is the main extracellular cation, but it's the balance between sodium and then potassium that affects resting membrane potential. So if potassium is out of balance, whether it is high or whether it is low, it will cause electrical disruption by affecting the resting membrane potential. In skeletal muscle, it causes muscle fatigue and weakness, either because the membrane becomes hyperpolarized, or because even if it's in levels of high potassium, it'll result in depolarization, but then there's a hard time resetting that membrane potential. So high or low, disruption in potassium balance affects the resting membrane potential. And when we get to the heart, this disruption in electrical conduction can actually lead to a heart attack and potential death. For potassium, potassium is going to control its own extracellular concentration by two mechanisms. The two main mechanisms that regulate potassium secretion are when principal cells in the collecting duct sense a high level of potassium, then they will increase secretion of potassium into the filtrate. The other mechanism is aldosterone. Adrenal cortical cells are directly sensitive to the extracellular concentration of potassium. When the adrenal cortical cells sense an increase in potassium concentration, then the adrenal cortex will release aldosterone. And aldosterone will reabsorb sodium, but it also increases potassium secretion. So for regulation of potassium balance is important because it maintains the resting membrane potential, and that affects the ability of cells to depolarize. And the main hormone that regulates potassium balance is aldosterone, which is secreted by the adrenal cortex and causes an increase in potassium secretion. Moving on to calcium and phosphate. So 99% of the body's calcium is in bones as calcium phosphate salts. So calcium is important because it builds healthy bones, but calcium is also important in regulating neuron action potentials. In general, we keep a very tight blood calcium level of 9 to 11 mg per deciliter. An increase in calcium causes inhibition of neurotransmitter release. So neurons and muscle cells are not able to conduct like they should. So maintaining the correct balance of ionized free calcium in the blood maintains proper neuron and skeletal muscle activity. So that's why it's important to regulate calcium. Now, how do we regulate calcium? Calcium balance is under exclusive control by the parathyroid hormone. We are constantly losing calcium from the blood, either used by neurons and skeletal muscles, but also if we're building bone. But calcium is always leaving the bloodstream. And so parathyroid hormone is going to regulate to bring it back up. The way that calcium is regulated in the blood is through parathyroid hormone. Parathyroid hormone is going to promote an increase in calcium level. It stimulates osteoclasts to break down the matrix, releasing calcium and phosphate to the blood. We'll talk about phosphate in just a second. For the kidneys, it will increase calcium reabsorption, and the small intestine will also increase calcium reabsorption. So this slide, it shows how parathyroid hormone regulates blood calcium levels. It increases osteoclast activity, increases calcium reabsorption in the kidney, and increases calcium absorption from the small intestine. And this, by maintaining this balance of blood calcium levels, neurons and muscle cells will be able to properly generate potentials. We're going to go back real quick to phosphate. This releasing calcium and phosphate, this phosphate is actually going to be secreted and used as a buffer. And by doing that, it will make sure that calcium doesn't rebind to the phosphate and will be available to stay in the blood. So it won't get resequestered back into bone. Chloride is the main anion in extracellular fluid. It is going to be reabsorbed passively with sodium in the proximal convoluted tubule. It's not under hormonal control, so it's not really, I don't think you'll be seeing this one again. Okay, so we are going to move on to acid-base balance. If you remember, pH is a scale that goes from 0 to 14, and 7 is considered neutral. Between 0 to 7, this is considered acidic, and from 7 to 14 is considered basic. Because of this negative number before the log, it means that the lower the pH, the more acidic it is, the more hydrogen ions. So at a pH of 0, we have 1 molar hydrogen ions. And when we go up to a pH of 7, the concentration of hydrogen ions is 0.00000001 molar hydrogen ions. You will not have to calculate and tell me what is the concentration of hydrogen ions, but you do need to understand that the lower the pH number, the more hydrogen ions there are. So hydrogen ions are what create an acidic environment. So if you increase the number of hydrogen ions, then that solution is becoming increasingly acidic and the pH is becoming lower. So if we look at solutions, solution A is going to be a pH of 4. Solution B is a pH of 6. And then we have solution A is 100 times more acidic than solution B. So why is it 100 times more acidic? That's where this log comes into play. For every value from like 4 to 5, 5 to 6, it is a factor of 10. So when we have a pH of 4 and a pH of 6, the difference between those two is 10 to the second. So 10 squared. So 10 times to the second, because there's 2 levels, 4 to 5 and 5 to 6. 10 to the second is 100. If the pH was 7, then solution A would be 1,000 times more acidic, which is 10 to the third, because there's 3 levels of difference, 4 to 5, 5 to 6, and 6 to 7. That's a brief review of pH. If you still need more practice, there are a lot of videos available that will discuss pH. With pH, it is only a measurement of free hydrogen ions. If they are sequestered in a compound like carbonic acid, which is this one, it does not count against the pH. Only the free hydrogen ions. So one of the things about a buffer system is it is going to be a system made up of a weak acid and a weak base. And the difference between a weak acid and a strong acid is the amount that it dissociates. A strong acid will completely dissociate. So if we put in 100 molecules of HCl, we would get 100 chloride and 100 hydrogen ions. If we put in 100 molecules of carbonic acid, we would get 90 that stay as carbonic acid, 10 hydrogen, and 10 bicarbonate. So that is kind of a primer, some background information on pH. Now we will go back to our material. So I just mentioned that pH is neutral at 7. However, our blood, its physiological pH is 7.35 to 7.45. So when we talk about acid-base balance, we're talking about arterial blood. So we'd be talking about arterial blood gas measurements. So this is an arterial sample that's taken, and then we'll measure things like pH the partial pressure of oxygen, the bicarbonate concentration, in order to assess health. But in the blood, for arterial blood, the range of a normal pH is 7.35 to 7.45, and that is a value that you do want to know. The reason why you need to know this value is to determine something called physiological alkalosis and physiological acidosis. Physiological alkalosis is when the arterial pH is over 7.45, and the physiological acidosis is when arterial pH is less than 7.35. So why do we care so much about pH? We have proteins. Our body is made up of proteins, so it's made up of structural proteins, but we have functional proteins that make our body work. They catalyze nearly every reaction in our body. Some things occur by diffusion, but for the most part, we need functional proteins. And if you remember back to last year, we had proteins that they have a primary structure, a secondary structure, a tertiary structure, and then something called a quaternary structure, where you have polypeptides that fold into each other. And the reason why these proteins fold in and they'll make a binding site for a substrate is because they have properties that attract certain parts to other parts. And one of those properties is going to be charge. So you'll have a negatively charged and a positively charged, and they'll come together and they'll stabilize that structure. When you change pH, the structure of the enzyme, that functional protein, can change, and then it's not functional anymore. So when we get down to 7.1, 7.6 or 7, we're really risking death because our body is going to stop functioning as it should. So our body is going to be very careful about maintaining acid-base balance, and this is a large priority for the respiratory and renal system. Okay, so for this slide, make sure that you understand that our samples are coming from arterial blood. Venous blood is slightly different because it has an increase in metabolic products, such as just metabolic acids and carbon dioxide. You also need to know these values. That alkalosis is when arterial pH is over 7.45, and acidosis is arterial pH is less than 7.35. So from this slide, you need to know these three values. Where do body acids come from? So we have already discussed that our normal arterial pH is 7.35 to 7.45. In a healthy individual, we are usually adding hydrogen through metabolism. And so we're usually in a state of declining pH. So things without intervention, our blood would become more and more acidic. There are conditions that make things alkalotic, but those are almost exclusively due to there's a someone's not healthy. It's a disorder. So in general, when we present examples, we usually start with pH is declining because that's what is going on the vast majority of the time. So why does our blood pH become increasingly acidic? The reason why is because we are adding hydrogen ions as byproducts of metabolism. When we break down protein, glucose, and fat, those generate acidic byproducts. So when we break down phosphorus-containing proteins, phosphoric acid is released. With anaerobic glycolysis of glucose, lactic acid is released. With a breakdown of fat, that is going to be broken down into fatty acids and ketone bodies. So metabolism generates metabolic acids. Metabolic acids increase the hydrogen ion concentration, leading to a decrease in pH, which, if left unchecked, we would not survive. So how do we regulate hydrogen ions? How do we maintain acid-base balance? Hydrogen ions are regulated by 3 systems: chemical buffer systems, respiratory centers, and renal mechanisms. Chemical buffer systems are the fastest. They are immediate, but they are dependent on how much weak acid or weak base you have available, because they tie up hydrogen ions, but they don't actually excrete them. So they're great because they're quick, but their capacity is limited. When we get to respiration, when we breathe out CO2, CO2 acts as an acid, so in effect, we are getting rid of an acidic product. With renal mechanisms, we have the ability to actually secrete hydrogen ions into the urine, as well as generate new bicarbonate. So have a more, what's called an alkaline reserve. So we'll start with chemical buffer systems. So chemical buffer systems always have a weak acid, a weak base, and sometimes these are called conjugate acids and conjugate bases. And then often there's something called a conjugate salt that's involved in the reaction as well. It becomes a product. But every chemical buffer has to have a weak acid and a weak base. That's how they buffer. Okay, so in these systems, when something is acting as a weak acid, it will release a hydrogen ion. And when it is acting as a weak base, it takes a hydrogen ion out of solution. So it's going to bond and tie up this hydrogen and take it out of solution. So weak acids release hydrogen ions. Weak bases accept them and tie them up. Okay, we went through this. For chemical buffer systems, there are 3 systems. For the bicarbonate buffer system, and I have a summary slide after we finish kind of discussing each one. There's a summary slide that will be helpful in helping you sort out what about these 3 chemical buffer systems do I need to know, and what is interesting, but maybe I will just reference later. Okay, so we'll talk about the 3, then we'll come to a summary slide where I will summarize the items that you truly need to know. So the bicarbonate buffer system is the important buffer system for extracellular fluid. So the plasma is really what we talk about. Yes, there's interstitial fluid as well, but it equilibrates with the plasma. But we are generally talking about the plasma because that's where these reactions take place. So the bicarbonate buffer system is the main chemical buffer for the extracellular fluid. It is going to be made up of a weak acid, being carbonic acid, and then a weak base, which is, in this case, is sodium bicarbonate, also known as baking soda. And then its conjugate salt is sodium chloride. If we were to increase the acid concentration, so in this solution, it's becoming more acidic, it will what's called drive the reaction to the right. So when we get a buildup of substrate, then it puts pressure on this reaction to go in this direction. We're going to discuss the bicarbonate system more in detail when we get to renal regulation. What this is showing is that it's important to have an alkaline reserve. So when we talk about regenerating, reclaiming bicarbonate, one of the reasons is so that we have more of this weak base available to interact with if we're adding in hydrogen ions. If we don't have enough bicarbonate to react with, we can't move the reaction. We can't accomplish it. You need both substrates. So maintaining enough bicarbonate in the plasma is very important for buffering the hydrogen ions in the plasma. That's what this is saying. The system only works if there is enough weak acid, weak base available to release protons. In our body, we're not going to have a deficiency of acid, because that's what we tend to be buffering. We tend to be producing a lot of hydrogen. So that is pretty much never the limiting component. What becomes limiting in this extracellular fluid chemical buffering system is the amount of bicarbonate. Again, sodium is usually readily available, but this component, the bicarbonate component, if we don't reclaim it, we don't regenerate it, we won't have enough bicarbonate to make this work. It's like trying to make, you need to make 24 cupcakes and you only have a cup of flour. You won't be able to make those cupcakes. So in our example, enough bicarbonate is the limiting factor in making the system work. The phosphate buffer system is the only true effective buffer in urine. It will have a little small role in intracellular fluid, but highlighted is the buffer in urine. Like all chemical buffering systems, we have a weak acid and a weak base. And what changes, if you can take a look, is the number of hydrogen ions between these two. This is not you don't need to know these reactions. For phosphate, you do need to know that it will be utilized in the urine, and we'll be going into that more when we get to renal regulation. And then just like all systems, it has a weak acid and a weak base, but you will not be expected to write out these formulas. Protein buffer system is the main intracellular buffering system. So we have our cells, we have a nucleus, we have lots of proteins. We have ribosomes. Those ribosomes are making polypeptides. We have just a lot of globular enzyme proteins that are in the cells. We have a lot of protein in our cell. Proteins are called amphoteric. Each amino acid can serve as both the weak acid and the weak base. Proteins are made up of kind of three areas. We have our group. that gives a protein its unique characteristics. Is it going to be acidic? Is it going to be basic? Is it going to be polar, non-polar? But it does not play a role in the buffering system. The two components that do come into play in the buffering system are we need a weak acid. The portion of the group that plays the role of weak acid is the carboxyl group. So the carboxyl group can function as a weak acid. If pH drops, this hydrogen becomes released. It gets released into solution. And then this part of the protein is going to carry a negative charge because it lost a hydrogen. So now it's slightly unbalanced charge-wise, but it's okay with a protein, they don't mind. Or if for our weak base, pH is decreasing, hydrogen ions are increasing, the amino group can act as a weak base, and it will take this hydrogen ion and it will form a bond. And if you remember way back into 1147 lab, some of the nitrogens have 2 holes and some have 3 for making bonds. And that's because nitrogen can transition between 2 and 3 bonds with other molecules. So nitrogen, this amino group, will make a bond with a hydrogen, and then it will also carry a positive charge now on this end. So it becomes a charged protein. For proteins, you need to know which part of an amino acid can serve as a weak base or a weak acid. So the amino group can accept a hydrogen ion, and that has the amino group functioning as a weak base. It will take this free hydrogen out of solution and it will increase pH because it's working as a base. The carboxyl group can be a proton or hydrogen ion donor, and so it is working as a weak acid. When the carboxyl group donates the hydrogen ion, it will then cause a decrease in pH. So you do need to know which group of an amino acid, and this works for proteins as well. So if you had amino acid, amino acid, amino acid, amino acid, amino acid, the amino group at one end and the carboxyl group at the other will be the ones that are acting as either a weak base or a weak acid. So it doesn't have to be a singular amino acid. In fact, it's not usually going to be a single amino acid. It's probably going to be a polypeptide. As I promised, we have a summary of chemical buffer systems. All chemical buffer systems are made up of a weak acid and a weak base. For the bicarbonate buffer system, carbonic acid is the weak acid, bicarbonate is the weak base, and it is going to be the main extracellular buffer. For the phosphate system, the main component is going to be phosphate. Dihydrogen phosphate is the weak acid. So this one has 2 hydrogens, which is why it is the acidic component. And then this is monohydrogen phosphate. So it's lost a hydrogen. And so this is the weak base. It is the primary system functioning in urine. For the protein buffer system, it is the most important buffer in intracellular fluid. It is called amphoteric in that its side chains act as weak acids in the case of the carboxyl group, or the side chain that works as the weak base is the amino group because it can accept a hydrogen ion. So you do need to know which side chain is a weak acid, which side chain is a weak base, and how they work. That the amino group is a proton acceptor. The carboxyl group is a proton or hydrogen donor, which is the definition of an acid and a base. We are now going to move on to the respiratory and renal systems. They do act more slowly than chemical buffers. So respiratory is in minutes and to hours, even days. I mean, like, it has a prolonged effect, but then in renal will take hours, days, and it can stay in effect for weeks. But the thing about these physiological buffering systems, respiratory and renal, is they can actually excrete acidic products. Or in the case of bicarbonate, you can actually, if you got alkalonic, you can excrete bicarbonate as well. So these can actually change the concentration of hydrogen and the concentration of bicarbonate. For this equation, you need to be able to write out the equation in as a chemical equation and in words. So we have carbon dioxide plus water, and then this can form carbonic acid, and carbonic acid dissociates into hydrogen ion plus bicarbonate. An enzyme is required to for this part of the reaction to occur. So to go from carbonic acid to carbon dioxide and water, or to go the reverse, requires carbonic anhydrase. So a enzyme is required at this step, but for carbonic acid, it will, if under the right conditions, dissociate into hydrogen ion and bicarbonate. For the respiratory system, the respiratory system can eliminate CO2. So it is going to control this reaction by removing CO2. CO2 always acts as an acid. And the reason why CO2 acts as an acid is that if you build up and you have a lot of CO2, it puts pressure on this reaction. So there's a lot of substrate. And that substrate is then going to combine with water, which is not going to be limiting under these conditions. We've reabsorbed enough water, we've got plenty of water. So if we build up CO2, water is not limiting, then that is going to cause a buildup of carbonic acid. And that means that more of this carbonic acid will dissociate into hydrogen ions and bicarbonate. For this reaction, hydrogen ion is a free ion. So we may work with this ion later, okay? But for now, we're talking about what happens when we build up CO2. And remember, only hydrogen affects pH. pH, by definition, is going to be inversely related to the concentration of free hydrogen ions. So as hydrogen ions increase, pH, the value is going to decrease and things become more acidic. Remember, a lower pH indicates higher acidity, higher hydrogen concentration. So how do we regulate hydrogen ions? So this first one says, okay, we are now in acidosis. So there is a high concentration of hydrogen. So acidosis is a high concentration of hydrogen. If CO2 is an acid, in order to compensate for acidosis, we need to get rid of CO2. Under states of acidosis, respiratory rate and depth are going to increase. So we can get rid of CO2 and in effect, get rid of the hydrogen ion. In a state of alkalosis, the hydrogen level is too low. We know that CO2 acts as an acid. So we want to retain CO2. So CO2 is getting produced by our cells. And if we can exhale less or less deeply, we will keep that CO2 retained in our body. It will act as an acid and it will restore pH balance. Hypoventilation causes respiratory acidosis. When we're not removing enough CO2, so we don't breathe enough, CO2 builds up, resulting in acidosis. When we hyperventilate, so we're breathing too much, we are exhaling all of our CO2, which acts as an acid, and that will cause alkalosis. We'll talk more about acidosis and alkalosis when we get to metabolic disorders. For now, be able to write this equation both in chemical format as well as in words. You should also be able to identify that the respiratory system works on the CO2 component of this equation. CO2 acts as an acid. So if you increase CO2, that will cause an increase in hydrogen ions, which leads to a decrease in pH. Normally, in acid-base balance, we're dealing with high CO2, high hydrogen that we want to get rid of. And so we want to decrease the CO2. So if our blood is becoming more acidic, we want to remove CO2 by breathing deeper or deep and faster to get rid of the CO2, and that drives this reaction this way. We are now going to discuss renal regulation. Renal regulation is very important because only the kidneys can eliminate metabolic acids. In respiratory regulation, we eliminate CO2, which does affect pH through this equation. However, we aren't directly excreting hydrogen ions. For the kidneys have two very important functions regarding acid-base balance. One is that active secretion of hydrogen ions is possible. The other, and probably the more important one, is to conserve or regenerate bicarbonate. So bicarbonate, if you remember, we have our glomerular capillaries, and then we have our proximal convoluted tubule. Okay, so bicarbonate is going to be in the filtrate. It's going to, as filtrate is produced, bicarbonate is going to be at that same concentration in the filtrate as it is in the extracellular fluid. So we are constantly losing bicarbonate through filtrate formation. Now, this is a problem because the tubules are impermeable to bicarbonate. So we cannot directly, the kidney tubules cannot directly reabsorb bicarbonate. Okay, so bicarbonate is going to be filtered out with the filtrate, and then we need to reclaim it, and so that it can be reused in the system. So in order to maintain alkaline reserve, the kidneys must reabsorb bicarbonate. Tubule cells are impermeable to bicarbonate. So bicarbonate is getting, it's going to be part of that filtrate coming through the tubule system, and we must reclaim bicarbonate or else we lose our alkaline reserve. But with the tubule cells being impermeable to bicarbonate, there's no transporter for bicarbonate. It has to go back, gain entry back into the body in a roundabout way by being converted to CO2. So the mechanism is coupled to hydrogen secretion. For every hydrogen secreted, a bicarbonate is reabsorbed. This is the first of three diagrams. The first step that you want to take in evaluating each of these three diagrams is how many bicarbonates are you starting with? And then how many bicarbonates are you going to end up with? In this figure, we start with one bicarbonate entering the filtrate. Okay, so we have bicarbonate that has come from the extracellular fluid, it's gotten captured with the filtrate, it is now collected into the proximal convoluted tubule, and this bicarbonate has entered the filtrate. Again, bicarbonate cannot directly transport across the proximal tubule. It has to get converted to carbon dioxide first. So we start with one bicarbonate entering the filtrate. It is going to combine with a hydrogen ion that does have a transporter. So hydrogen ions can be transported, and it will get converted to H2O and carbon dioxide. The carbon dioxide can diffuse, and it will combine with water using carbonic anhydrase to become bicarbonate again, and then bicarbonate can get transported to the peritubular capillary. So in this system, we start with one bicarbonate. It is going to get transformed into CO2, and then it gets transformed into carbonic acid, and then it gets returned as bicarbonate. So we start with one bicarbonate and we end with one bicarbonate. Okay, so we start with a bicarbonate in the filtrate. We end up with a bicarbonate in the peritubular capillary. So we have reclaimed one bicarbonate that would have been excreted. Okay, so we've saved it. At first glance, it looks like, cool, we have secreted a hydrogen ion and we can get rid of that. But in reality, this hydrogen ion has now become a part of water. And this water is going to get reabsorbed. So we've reclaimed this bicarbonate, but we haven't excreted a hydrogen because the hydrogen gets transformed into water and then the water gets reabsorbed, and that hydrogen, even though it's now in water, which eventually it can get cleaved and off. So that hydrogen ion hasn't gotten excreted as it looks like at the first. So the question is, what do you need to know about this graphic? You need to be able to identify that the purpose of the process being displayed in this graphic is bicarbonate reclamation. Bicarbonate, which is HCO3 minus, is saved from excretion or rescued from excretion. And returned to the extracellular fluid. However, hydrogen ion balance is unchanged because water is reabsorbed. So we haven't really made things better, but we have prevented them from getting worse. So how can you tell that this graphic is about bicarbonate reclamation? You can tell that this is about bicarbonate reclamation because we have start with the filtrate, and here is the bicarbonate, and then you end up with a bicarbonate following the pathway through the cell and then back to the peritubular capillary. So if you trace the bicarbonate, it is going to return to the peritubular capillary. The next two pathways are going to talk about the two mechanisms to generate new bicarbonate. One pathway will use the phosphate buffering system, and then the other pathway will use glutamine metabolism. This graphic is showing the phosphate buffering system in the urine. The first thing to note is find bicarbonate in the lumen. In the last graphic where we reclaimed bicarbonate, there was bicarbonate in the filtrate. If you look at this graphic at the filtrate, we're not talking about reclamation of bicarbonate. There's no bicarbonate to be found in this process. Okay, so when you're identifying, is it bicarbonate reclamation or are we making new bicarbonate? Look to see, are we starting with any bicarbonate in the filtrate? In the next two pathways, we will not be starting with bicarbonate in the lumen. In the graphic, of course, it is present, but not in the pathway that we're talking about. Then how do you know that it's the phosphate pathway? Well, it says right here, we're going to use the phosphate buffering system in the urine. So phosphate, this is monohydrogen phosphate. It can accept a hydrogen ion that has been secreted into the filtrate, into this the lumen, and now it combines to form dihydrogen phosphate, and it is excreted to the urine. So in this process, we truly lose a hydrogen ion because it is captured by the phosphate buffering system and excreted. Unlike in the previous pathway where it becomes a component of water that is reabsorbed, this hydrogen ion is going to get excreted as part of dihydrogen phosphate in the urine. So we get one hydrogen that is truly secreted. While we do that, we also get a new bicarbonate. Both carbon dioxide and water are going to be able to diffuse at will. With carbonic anhydrase, it creates carbonic acid, which then dissociates into hydrogen and bicarbonate. The hydrogen is going to be actively secreted. The bicarbonate is going to be a new bicarbonate placed into the extracellular fluid. So when evaluating this graphic, there are a few things to look at. What system are you using? You are using the phosphate buffering system of the urine. So look for phosphate in the filtrate. It is going to combine a hydrogen ion, so we actually excrete a hydrogen ion, and it is going out to the urine. So that hydrogen ion is excreted. At the same time, if you look over here in the circulatory system, you'll see the word new, meaning that a new bicarbonate has been generated. Okay. So phosphate buffering system, look at what we're using, phosphate over here. You lose one hydrogen ion out into the urine, and one new bicarbonate ion is generated. The third pathway that we are going to discuss is the glutamine pathway. Just like with the previous two graphics, start at the lumen. What are you starting with? In the glutamine pathway, you will be starting with glutamine. Glutamine is then transported into the cell, and then it is essentially split. It's going to be split into ammonia and bicarbonate. The ammonia is going to be transported out, and then it will be excreted in urine. Even though this is positively charged, this is not secreting hydrogen ions. It occurs during the process of splitting the glutamine. However, it does generate two new bicarbonate. So we have two new bicarbonate over here. Okay. So if you look at all three pathways, the first pathway that we looked at was the reclamation of bicarbonate. It didn't create any new bicarbonate, but it saved one from being excreted. So you had one in the filtrate and you ended up with one in the capillary. So there's nothing that's new. It's reclaiming bicarbonate from the filtrate so that we don't lose alkaline reserve in the extracellular fluid. In the phosphate pathway, a hydrogen ion is secreted, and at the same time, a bicarbonate, a new one, is generated. For the third, the glutamine pathway, We're not actively secreting hydrogen, but we are generating 2 new bicarbonate. So those are the key take home from these 3 graphics. You hopefully will study these 3 graphics because you will have questions on them, such as, are they reclaiming bicarbonate? Are they generating new bicarbonate? Is secretion of hydrogen taking place? So there will be a question for each of these graphics. That brings us to the end of today's lecture. We will pick up on next week with abnormalities of acid-base balance.